We rarely get to perform electrophysiological recordings within the brains of humans. Instead, we need a method to non-invasively measure neural activity. One of the most common methods for measuring neural activity in humans is functional magnetic resonance imaging, or fMRI for short. First, let's talk about structural MRI, which doesn't measure brain activity, but rather shows us the anatomy of the human brain. Here, we have an image from structural MRI of the brain. This is a coronal slice, so a plane vertically through the brain. You can see that some parts are darker and some lighter. This corresponds to gray and white matter, cell bodies versus axons, discussed when we looked at slices of actual brain tissue. And of course, you can see the same sulci and gyri, folds, and bumps that we saw when looking at the brain surface or tissue slices. The details of these grooves may vary slightly from person to person, but overall they are important landmarks used to identify specific parts of the brain in fMRI studies. MRI data is acquired one 2D image or slice at a time, but you can take a stack of these 2D images and slices in different planes and combine them to form a 3D model of an individual's brain structure. There are certain types of questions you can ask just with this structural data. For example, you can look at the size of a particular brain area or the thickness of specific parts of the cortex and see how that relates to behavioral variables, including performance on a specific task, life experiences, or diagnosis or severity of behavioral symptoms in some neurological disorder. Most of the human brain studies you see coverage of in the press are probably functional MRI or fMRI studies. fMRI gives us an indication of neural activity in different brain areas. This measurement is very indirect. It reflects the relative proportion of oxygenated versus deoxygenated hemoglobin, which is related to blood flow, which is modulated by neural activity in a particular segment of brain tissue. The signal measured in fMRI is called the BOLD, or blood oxygen level dependent signal, and this signal is compared between two conditions. So, for a visual area, you might compare the BOLD signal when a person is staring at a blank screen versus some visual stimulus. And you would expect to see a larger BOLD signal in a variety of areas involved in visual processing when the visual stimulus is present on the screen. Here, we see the change in BOLD signal in the occipital lobe reflecting visual stimulation, with color representing the statistical strength of the activation superimposed on the structural MRI showing patterns of gray and white matter. The electrophysiological techniques we talked about directly measure the electrical activity of individual neurons. fMRI, in contrast, is indirectly measuring the activity of neurons within a small volume, less than a cubic millimeter, depending on the power of the magnet. But even that tiny volume will contain tens of thousands of neurons. Fortunately, the spatial grouping of neurons with similar response properties, first into brain areas and then at an even finer level of visual neurons with similar receptive fields being grouped together, for example. This grouping means that we can still see selectivity even when averaging over these large numbers of neurons. You can use fMRI to very clearly visualize the cortical maps that we talked about when we covered the visual system. In this example, visual stimuli are presented in different parts of space, and the BOLD signal throughout primary visual cortex is compared to the case of no visual stimulus. Here, locations in space are color-coded to match the parts of visual cortex that are active in response to visual stimuli appearing at that location. You can see that the representation of the center of the visual scene, where we have the highest resolution vision, is enlarged compared to how much cortex is devoted to representing stimuli in the periphery. And you can see that there's a systematic mapping of visual space onto the cortex. Locations in space that are next to one another activate parts of cortex that are next to one another. This relationship between the location of a visual stimulus in space, and hence where light bouncing off that stimulus will hit the retina, and the location of the neurons it drives in visual cortex is referred to as retinotopy, or retinotopic mapping. This type of mapping illustrates that the spatial resolution of MRI is finer than a brain area, even though it's averaging over tens of thousands of neurons at a time.